Okay. Thanks, Conchita, for the introduction. So thank you very much to all of you for being here today. I want to say first that I would have liked to have given this talk earlier, like soon after I arrived here in November. The thing is that I didn't have that many results to show you, so I had to wait until now to introduce myself to all of you. So I'm going to talk about this project called BRY'O'MICS. This is the short name for a project that aims at understanding the mechanisms or gaining more understanding on the mechanisms underlying heavy metal accumulation and tolerance in terrestrial bryophytes. So this project has been funded by the European Union through the program Horizon 2020, and more specifically through the Maria Sklodowska-Curie actions, which are grants that are awarded to individual researchers in order to enhance their research career. So I'm just showing here a little bit of information about the project. It is a global fellowship, which means that lasts over 3 years. It started in July 2016, and it'll finish, unfortunately, August 2019. So since it's a global fellowship, I spent 3 years collaborating with researchers from 3 different countries between the United States and Europe. So my first stop was in the United States, where I spent the first 2 years of the grant working under the supervision of Christina Richards. I did the bulk of my experimental work there, including sample collection and lab work. Christina works mainly in studying how non-model plants adapt to environmental stress and environmental change, especially applying genomics and epigenetics tools. Then I did a short stop at the University of Florida, at the University of Vienna in Austria, where I did a small part of the experimental work that I'm not going to talk about here about it today. And finally, my last stop was at the EBD, where I arrived in November, and I joined the Herrera lab, mostly under the supervision of Conchita Alonso and Carlos Herrera. You all know what their work is about. They mainly study plant-animal interactions, and they also study natural populations from genetics and epigenetics perspectives. So here, I was supposed to do pretty much all the analysis of the results that I obtained doing the lab work, and of course, writing papers. So the idea for this project arose when I was finishing my PhD, which basically consisted in an experimental theoretical evaluation of the use of the MOS technique to monitor atmospheric heavy metal deposition. So for those of you that don't know what the MOS technique is, it basically consists in collecting samples of terrestrial bryophytes in different areas that we want to study, measuring the concentrations of heavy metals in their tissues, and trying to reach conclusions about the state of the environment in those specific areas. So this technique has been used especially in Europe for over more than 50 years. And there are different researchers that have been working all across Europe, collecting samples, performing surveys every 5 years, and collecting samples of bryophytes in order to build contamination maps, such as the one that I'm showing here. However, when I was finishing my thesis, I started to think about how these quite simple plants were able to tolerate and accumulate such high levels of heavy metals in their tissues. And the truth is that I started to look at the literature, and I didn't find a lot of information about, for example, the heavy metal dynamics inside the plants for bryophytes, or the evolutionary consequences of accumulation and tolerance in bryophytes, or even the ecological role of heavy metal hyperaccumulation, for example. The truth is that the bulk of the literature was addressing all these different kind of issues in vascular plants, especially in the angiosperm group. And this literature revealed to us that, for example, heavy metal homeostasis happens at 2 different levels in vascular plants. The first level would be the whole plant level, by which different accumulation and tolerance strategies imply regulation of, for example, root uptake of heavy metals, also regulation of the transference of heavy metals between the roots towards the above ground tissues of the plants, also regulation of or controlling the amount of heavy metals that are actually translocated towards deciduous tissues or less physiologically active tissues or kind of cells. And finally, of course, limiting or controlling the amount of heavy metals that are transferred into seeds and therefore into the offspring. However, this kind of regulation cannot happen in bryophytes, basically because their body structure is completely different to that of vascular plant. It's not structured in the same way. And I'm going to talk a little bit about it a little bit after in the presentation. So the second level of regulation for heavy metal accumulation and tolerance happens at the cell level, where, for example, the cell wall constitutes the first checkpoint for heavy metals trying to enter the cell. So there are different compounds in the cell wall that are able to bind heavy metals and prevent them from entering the cell cytoplasm. The second checkpoint would be the plasma membrane, where there are different membrane transporters that are able to pump heavy metals inside and outside the cell, depending on the needs of the cell. And at the cytoplasm level, the cell can produce different organic compounds that are actually able to bind and chelate those heavy metals and to transfer them into other cell compartments, such as the vacuoles, where they can be sequestered, and therefore they will be less toxic than in the cytoplasm itself. So at an evolutionary level, heavy metal toxicity imposes a very strong selective pressure in plants, especially because they cannot run away from heavy metal contaminated places. And therefore this has led to the development of genetically distinct races, or what we commonly know as ecotypes, especially in vascular plants, such as species like the ones that I'm showing here. And finally, about the ecological roles, there are different hypotheses that are more or less supported by experimental evidence that would explain why plants would like to hyperaccumulate some heavy metals. However, there is a considerable gap of knowledge in all these issues for bryophytes. There's not much literature, despite the fact that some bryophytes species are really capable of accumulating high levels of heavy metals in their tissues and tolerating those high levels of heavy metals. And even that some species, such as the ones that I was highlighting here in red, are even specialized to live in heavy metal contaminated places. So now I wanted to tell you a little bit why I do think that bryophytes are really interesting plants. And the first reason is because they are in a key phylogenetic position within the land, they are in between green algae and vascular plants, and they have been among the first plants colonized in the land environment, and therefore they have been able to develop all the adaptations needed for the transition into land. Bryophytes are divided in 3 different groups: the liverworts, the mosses, and the hornworms. My work mainly focuses in the moss group. The second reason is that despite the fact that these plants are often overlooked because they are very tiny, they are a highly diverse group. In fact, there are about 24,000 different bryophyte species, considering all 3 groups together. And they therefore are the second most diverse group of land plants, of course, after the angiosperms group. And the third reason is that bryophytes can colonize almost any ecosystem on Earth, except for the marine environment. They can be found in water bodies and different environments in land. And therefore, they are capable of dealing with a wide variety of abiotic stresses. For example, they can be found in polar areas or alpine areas around the world, where they have to cope with really low temperatures and high levels of UV radiation. They can also be found in sand dunes, such as here, or in deserts around the world, when they have to withstand very high temperatures and also low levels of water availability. We can also find them being part of these pre-important communities called the biological soil crusts. These are very important communities that are found in drylands around the world. And just as a final picture, I wanted to show you that they are able to thrive even in geothermal vents, like environments such as stream as those. Just so that to give you a picture of how resilient these plants are, I wanted to show you this news, which is from back 2013, in which several researchers were able to get some moss tissue that had been buried for over 400 years under a glacier. When the glacier retreated, they were able to collect these tissues, supposedly dead, and they were able to regenerate new living tissue in the lab without even using very special culturing techniques. And these plants are capable of doing all this despite the fact that they are quite simple from a morphological and an architectural perspective. So these plants are non-vascular plants, and they have a haploid-dominant generation called the gametophyte, which has mainly two life stages. The young life stage would be called the protonema, and it's basically a filament of cells that at some point further divides to form a bud, and that further develops into the leafy plant, which is called the gametophore. So the gametophore, it's very small. It can be from only a few centimeters for a few millimeters to a few centimeters in size, and it's quite simple. And despite it looks that it has a structure a little bit like a vascular plant, it doesn't. I mean, it has a stem, it has leaves, it has root-like structures that are not actually roots. For example, one interesting point is that their cells are mostly one cell layer thick, and they lack the typical protective cuticle. So all their cells are really in direct contact with the external environment. And another thing that you couldn't see here is they have these structures called rhizoids that, even though they look a little bit like roots, they don't work like roots. They don't have an homologous function. Their function is mostly anchoring the plants in the substrate because water and nutrient absorption mainly happens throughout the whole body of the plant in bryophytes. So bryophytes are poikilohydric, meaning that they have a very low capacity of self-internal regulation, and so they depend on the external conditions for their functional activity. The diploid generation is called, it's this sporophyte that looks like it has a stalk and a capsule that contains, would eventually contain the spores. And it's always dependent and attached to the mother plant. It's nutritionally dependent and always attached to the leafy gametophore that you can see here. So bryophytes can undergo both sexual and asexual reproduction. When sexual reproduction happens, they form this sporophyte that eventually will release the spores into the environment, and that will germinate and generate new protonema that would also generate new leafy plants. But also asexual reproduction is pretty important for bryophytes. In fact, some species mostly rely on asexual reproduction for their dispersal. And they can do it either by producing specialized vegetative propagules, like you can see here these kind of cells that are detaching from the protonema. These are called gemmae, or even broad cells that are specialized structures that they detach from the protonema, and they are released in the environment so that they grow into a new individual. But another important characteristic of bryophytes is the fact that all their cells in their gametophores are totipotent, meaning that they can regenerate a new plant even from a tiny, small fragment of a leaf or a root or a stem or anything. And this is a pretty important characteristic for growing them and culturing them in the lab. So now that you know a little bit more about why I'm so keen on bryophytes and why I like them so much, and a little bit more about the general scope of BRY'O'MICS, I wanted to tell you a little bit about the specific questions that we wanted to answer with this project. And the first one is to find out whether there is phenotypic variation for heavy metal accumulation and tolerance in bryophyte species. The second one would be to check what are the genes or the molecular pathways involved in heavy metal accumulation and tolerance in this species. And finally, to check whether populations of bryophytes are genetically or epigenetically differentiated, and whether those differences are related to the levels of heavy metals that they experience in the field. So in order to answer all these questions, we designed a pretty multidisciplinary project in which we combined information from different disciplines like chemistry or microscopy, ecophysiology, and finally, of course, the omic tools. Today, I'm only going to focus on the results that we obtained for these different parts of the project. And for this, we were working with two different terrestrial bryophytes. The first one is called Scopelophila cataractae, and it's a species that has a pretty broad geographic distribution worldwide. It can be found in different places around the world, but its distribution is pretty disjunct, meaning that it can only live in heavy metal enriched substrates. For this reason, I'm going to refer to this species as the heavy metal specialist from now on. It can inhabit habitats like this one, which is the mine in which I collected all the samples for this experiment. This species is a copper hyperaccumulator, so it can reach levels from 1 up to 3% its dry weight in copper. And it is dioecious, meaning that it has separate, male and female plants are separated. And at least the populations that we were working with reproduce mostly asexual. No sexual reproduction have ever been observed in the field for these populations. The second species is called Serotonin Porters. You can see it here, and this is one of the most cosmopolitan moss species. It can be found in many places around the world, and it can be found both in disturbed and undisturbed areas, meaning that it can colonize heavy metal-enriched places and also pre-pristine areas. For this reason, I'm also going to refer to this species as the facultative metallophyte here. Serotonin is also dioecious, but as opposed to Scopelophila, it frequently undergoes sexual reproduction, mostly, but it can also reproduce asexually, of course. So this is a little bit the design of the sampling that we did. We collected the samples of both species in North Carolina in the United States. Scopelophila, we collected it in a mine site, and we collected 4 populations that were growing in a gradient of contamination. So in the populations that were collected in the center of the mine were supposedly exposed to high levels of heavy metals, whereas the populations in the edges of the mine were growing under lower levels of heavy metal. I'm always going to refer throughout the presentation to populations in the center and to populations in the edges of the mine so that you can pretty much understand where they come from. For Serotonin, we were only capable of collecting one population of this species in an urban area in Durham in North Carolina, but we also added to this study another population. This is actually only one population, although for simplicity I'm going to refer to it as 2 populations. But this is one population that was already growing in lab conditions, but male and female plants were growing separately in different places. So we could grow them separately and study them separately. So in addition to the plants in the field, we also collected soil samples right underneath each of the plant populations in order to really check whether they were actually exposed to different levels of heavy metals in the field. And before going ahead and showing you the experimental design in the lab, I wanted to tell you a little bit about the results of the concentrations of heavy metals in the soils and in the plants from the field for Scopelophila cataractae, from which we had different populations in the field. So here you can see the concentrations of copper for each of the 4 sites in which we collected the populations. So the red ones are the sites in the center of the mine, and the green ones are the sites in the edges of the mine. And you can see that the concentrations, actually, at least for copper, they are higher in at least one of the sites from the center of the mine. I also wanted to highlight the fact that this site 4 in the edges of the mine showed very low levels of copper in comparison with the other populations. However, when we look, so they were actually exposed to different levels of copper. However, when we look at the concentrations of copper in the plants that were collected in the exact same places, we see that this pattern that we observe here is not actually represented in the plants. So it looks like plants are not accumulating at least copper in a proportional way as copper is present in the environment. So with that, I wanted to show you a little bit what happens or what plants can do when they are exposed to heavy metals in the environment. If you see here the concentration of heavy metals in the soil and the concentration of heavy metals in the plant, plants have different strategies in order to take up these heavy metals. So the first one would be to take up heavy metals at a in a proportional way in comparison to the levels in the soil. In this case, we would be talking about indicator plants. However, they can also maintain very low concentrations of heavy metals in their tissues, whereas the concentration in the soil or in the medium are pretty high. In this case, we will be talking about excluder plants. And finally, they can reach really high concentrations of metals in their tissues, whereas the concentrations in the medium are still pretty low, in which case we will be talking about hyperaccumulators. So if we calculate the bioaccumulation factor, which is it's a simple number that results from dividing the concentrations of metals in the plant by the concentrations of metals in the soil. If the bioaccumulation factor is close to 1, we will be talking about indicator plants. If it's lower than 1, we will be talking about excluders. And if it's higher than 1, we will be talking about hyperaccumulators. So going back again to the graphs that I showed before, here you can see the bioaccumulation factors for all these 4 populations. And in this case, we could be saying that populations 1 and 2 are acting as excluders, whereas population 3 is acting as an indicator, and finally, population 4 is acting as a more or less hyperaccumulator. So this would suggest that maybe these populations have different accumulation capacities in the field. When we look at the data for cadmium, again, I'm showing the total cadmium concentrations for each of the soils and for each of the plants, and we can see a similar picture where concentrations of cadmium are higher in the center of the mine and lower in this population 4, in this side 4 from the edge of the mine. And in this case, both populations 1 and 3 would be acting more or less as indicators, whereas populations 2 and 4 would be acting as cadmium hyperaccumulators, especially population 4, in which concentrations in the plant reach 8.4 times the concentrations that we find in the soil. So going back to the experimental design that we did in the lab, we basically collected all 4 populations from Scopelophila and grew them for several months in the lab in soil pots. We also did the same with the field-collected population of Serotonin, but then we were able to sterilize field-collected tissue and grow it into in sterile conditions in the same way that the other populations were growing already, like male and female plants from the other population. So after they were grown for several months in the laboratory, we were able to perform the heavy metal treatments, and we basically did 1 control, 1 copper treatment, and 1 cadmium treatment for all populations in this study. Obviously, the concentrations of treatments for Scopelophila were higher than for Serotonin because they have different tolerance capacities. So we couldn't use the same concentrations. And once the experiments were over, we basically collected all plant tissue and tried to measure tolerance by measuring plant performance in terms, on the one hand, lipid peroxidation of the plasma membranes, which is basically a proxy for the level of oxidative stress that these plants are undergoing under the different treatments. And also we measured growth. Growth was measured in terms of colony size for Serotonin and of plant size for Scopelophila. And I'm explaining this. So when plants of Serotonin were grown in petri dishes in sterile conditions, they form protonymal colonies like this. So we basically measured growth by subtracting the final size of the colonies at the end of the experiment minus the initial size of the colonies. And for Serotonin, since they generated in soil, they generated gametophores, we basically measured the length of the plants and the length and the width of the leaves as measurements for growth for this species. In addition, in order to try to evaluate their accumulation capacities, we measured total concentrations of both cadmium and copper in the tissues. And finally, we did an experiment of gene expression where we used RNA-seq to evaluate gene expression changes between treatments. So we used these both measurements in order to answer the first question that I was talking about before, to find out whether there is phenotypic variation for heavy metal accumulation and tolerance. And I'm going to start showing you the results for the heavy metal specialist. And here I'm just showing the concentration of MDA. MDA is a product of the lipid peroxidation of the plasma membranes, meaning that the higher the concentration of this compound in the cells, the higher the level of oxidative damage that these plants are suffering. So this is for the 4 populations of Scopelophila. We have the control treatments, the copper treatments, and the cadmium treatments. So for cadmium, we didn't see a significant increase in the level of MDA when compared to the controls. But we did see a significant increase in the level of NDA for the copper-treated plants. However, this increase was pretty similar in all 4 populations. Therefore, we can conclude that there is no phenotypic differentiation in tolerance for copper in terms of oxidative damage. However, when we go to the growth measurements, as I was, before getting into this graph, I wanted to say that the only trait that responded to the copper treatment or to any of the heavy metal treatments was the leaf length, not the plant length, not the leaf width, only the leaf length. So those are the results that I'm showing here. So here you can see the length of the leaves of the 4 populations in the control and in the copper treated plants, and you can see that there was a significant effect of the copper treatment in the length of the leaves, but only for populations, for plants from populations 1. And even though this effect was not significant for population 4, the trend is pretty similar to what happened for population 1. So we could actually say that there is phenotypic differentiation for copper tolerance in this species in terms of growth. A similar picture happens when we look at the cadmium data. This is the same leaf length in all 4 populations in control and cadmium treated plants, and you can see that the patterns are pretty similar. In this case, the differences are not statistically significant for population 1, but they are for population 4, but the patterns are still pretty similar. So again, we could suggest that there is phenotypic variation for cadmium tolerance across these populations of Scopelophylla. When we look at the accumulation capacities, here I'm showing the total concentration of cadmium in control populations and in control plants and in treated plants for all populations, and the same for cadmium. And what you can see is that the concentrations of both metals in the treated plants clearly increased, really like 3 to 4 orders of magnitude with respect to the controls. However, when we look at the data for copper, we don't see any differences in the capacity of the plants to accumulate copper. But when we look at the cadmium data, we see that population 4, that originally in the field was hyperaccumulating cadmium, it has also a higher capacity to accumulate cadmium in lab conditions under common conditions. So with regards to the data for the other species, the facultative metallophyte, I'm showing the same kind of graphs here. So it's basically the concentration of MDA in the field collected population, males from population 2 and females. And you can see the different treatments, control, copper, and cadmium. In this case, copper didn't have a significant effect in oxidative stress for these plants, but cadmium did, and it did differently across all populations. So we saw a significant increase in the level of oxidative damage for the field collected plants and for the males from the population 2. However, we didn't see such increase for females from population 2. This suggests again that there is phenotypic differentiation for cadmium tolerance in these plants. If we look at the growth measurements, here I'm showing the colony growth for all field males and females in control and copper treated plants, and you can see that copper had a significant effect, significantly reduced growth in population that was already growing in the lab in a similar way in both males and females. However, it didn't in the other populations. So again, yeah, there is phenotypic variation for copper tolerance in these plants, whereas for cadmium, you can see the same kind of graphs, and the effects are a little bit different. So now population collected in the field is significantly affected by cadmium, and both females and males are also significantly affected by cadmium, but in a different manner. So males are actually more sensitive to cadmium than the other 2 populations. So yeah, we could conclude again that there are phenotypic differences for cadmium tolerance. In terms of accumulation, there's a similar picture to what happened with Scopelophylla, where the concentrations of copper and cadmium are shown for control and treated plants from the 3 populations. And you can see, well, the statistical tests are showing it here that there are no differences in accumulation capacities for these 3 populations. So just to summarize a little bit of the results that we found in this part of the work, yes, we did, for the copper specialist, or the heavy metal specialist better, we did find phenotypic differentiation for heavy metal, for copper and cadmium accumulation. And these differences were associated to the levels of heavy metals in their native environments, where populations 2 and 3 were more tolerant than populations 1 and 4 that were collected in the edges of the mine. And we did find phenotypic differences for cadmium accumulation, but not for copper. For Serotonin Porters, we observe significant differentiation for cadmium and copper tolerance, but sexual dimorphism for cadmium accumulation, for cadmium tolerance, which is something pretty interesting, and we want to look really deeper into this, and no differentiation for any of the metals accumulation. So now, in order to answer our second questions, what are the genes and the molecular pathways involved in metal accumulation and tolerance, we did an RNA-seq experiment, and the experimental design is the following. We couldn't do the analysis in all the plants, in all the samples that we included in the previous experiments, basically because of budget constraints. So what we did was selecting the control and copper-treated plants from both species, and in case of Scopelophila, we selected population 3 from the center and population 4 from the edges, and in case of Serotonin, we selected all 3 populations. So the first thing that I wanted to show you here, I'm going to show first the results for Scopelophila, the metal specialist, and this is a multi-dimensional scaling plot of the gene expression results. So what you can see here is that each pair of samples are separating according to the changes in gene expression across all genes that were differentially expressed in these samples. And so the farther apart the samples are from each other, the greater the differences in their gene expression profiles. So the first thing that I wanted to highlight here is the fact that the 3 replicates, I didn't say it, but we analyzed 3 replicates per population in treatment. And the 3 replicates cluster pretty well with each other, which is something nice in an RNA-seq experiment. And then when we look, we can also see that the treatments are more or less separated throughout the first dimension of the plot, with the controls both towards the right of the plot and the treated plants towards the left of the plot. What I also wanted to highlight here is the fact that control and treated plants from population 4 seem to be closer than control and treated plants from population 3 that was originally in the center of the mine. And this would suggest that genetic differences for these populations, that gene expression differences for this population are actually greater than for the others. If we look at the differentially expressed transcripts, we can see that there are a bunch of up and down regulated transcripts in the copper-treated plants when compared with each of their controls. And in fact, it is true that for population 3, the number of differentially expressed transcripts was way greater than for population 4. If we look at this very same data, but from a different perspective, here I'm showing the upregulated transcripts in population 4 and 3 and downregulated transcripts in population 4 and 3, you can see that the number of common up or downregulated transcripts is pretty low in comparison to the population-specific up and downregulated transcripts. So the idea now would be to look a little bit further in detail into the functions of these differentially expressed transcripts, the population-specific and of course also the common ones, to get an idea of what's going on in these metal-treated plants. So in addition to this, we did a gene set enrichment analysis, which basically consists in clustering genes by their gene ontology terms, and then looking, trying to check whether those clusters of genes are differentially expressed in the control plants when compared to the treated plants. So this would mean if they are differentially expressed, either up or down regulated, this would mean that this cluster of genes would be coordinately up or down regulated in the treated plants. And what we found here is that there were 133 clusters that gene clusters that went down regulated for population 3, and a bunch of them that were both up and down regulated for population 4. This is just very preliminary results, but if we look at the top 10 differentially expressed gene sets in those clusters, in the ones that I was showing here, we can see the gene ontology terms that are associated with those gene clusters, and we can see that they are kind of similar in both populations, but these are only the top 10. The idea is to look further into the specific genes or transcripts that are into these gene ontology terms and really get more into their function. So the picture for the other species, the facultative metallophyte, is a little bit different. Here I'm showing the same MDS plot as for Scopelophila, but here we can see that again, well, the replicas cluster pretty well, but the treatments do not separate. That's pretty interesting. And what we can see is that there is a clear separation in the gene expression profiles between male and female plants, which is interesting. But yeah, it wasn't what we were expecting. So when we look at the number of differentially expressed transcripts, we see that for the field collected population, there are a bunch of differentially expressed transcripts. This is also true for females from the other population, but nothing happened in males from that population, which is pretty interesting. And for that reason, after seeing this, we ran again I wanted to say that when we collected this plant in the field, we sexed the plants. So we basically looked for male and female gametangia in those plants, and all the sexually expressed plants were females. So potentially, the plants that we were working with in this population in the lab were females. And this makes sense given that their gene expression profiles respond, well, they respond in a similar manner to these copper treatments and completely different from the males. Looking at the MDS plot only considering the female populations, we see that now both treatments are separated across the first dimension. So this is a little bit more the picture that we were expecting. We're looking at the same data that I showed before. Again, the number of commonly up or down-regulated transcripts between the two populations was very low, and so most of them were population-specific responses. And it's interesting the fact that there are two transcripts that were upregulated in one population and downregulated in the other one. When doing the gene set enrichment analysis again, we observed a few less differentially expressed gene sets for these two populations, but here we found more upregulated gene sets, whereas for the other species we found more downregulated gene sets, which is also kind of interesting. So again, just to show you the top 10 differentially expressed gene sets, these are the gene ontology categories to which they pertain, and these, I'm showing them in red because they were upregulated gene ontology terms, and you can see that there are like gene ontology terms related to metal ion bonding and stuff like that. It's interesting, and it was expected. And these are the only two gene ontology terms among the top 10 that were downregulated in these two populations. So next steps would be to refine the differential expression analysis. We would really want to look into the differentially expressed genes that could be involved in all these mechanisms that I talked about at the beginning, that would be probably more applicable to the Bryophytes group instead of the whole plant regulation level. And also, we already have data about DNA sequencing for genetic and epigenetic analysis. We haven't been able to analyze it yet. We were having problems with that pipeline. But yeah, the idea is to get information about genetic and genetic differentiation among these populations. And we included the epigenetic dimension here because there is accumulating evidence that environmental stress can lead to epigenetic changes in plants that would eventually lead to phenotypic variation through regulation of gene expression, for example, that you can see here. And so this is pretty interesting for plants that are mostly asexually reproducing, because potentially they would have more limited genetic variation, and this epigenetic variation could provide an additional layer of phenotypic variation for these plants. That's why we were so interested in this part of the project. And near future steps would be, for example, studying heavy metal, the heavy metal fate inside the plants at the cellular level, like where are the heavy metals deposited in the plants, because that will give us an idea of the tolerance mechanisms. To do experiments with more species, and especially with more metals and metal combinations, because of course the responses of the different plants to single metals or metal combinations might be completely different. and study the cell wall. So the cell wall of bryophytes, it's a pretty interesting trait because it's the first barrier for the heavy metals trying to enter the cells. And we know that the cell wall structure changes when plants are exposed to heavy metals. So studying this structure would be very interesting in these plants. And I would like to finish the talk just with the acknowledgement part to all the people from different research groups that were involved in the lab experiments or in conversations about the data analysis, and especially the funding sources that allowed me to perform this project. And with that, I want to thank you all for your attention.